UConn research boosts microalgae protein for feed. UConn scientists have developed a method to raise protein content in microalgae by up to 25%, advancing sustainable protein production for both human diets and animal feed. Their findings, published in Biomass and Bioenergy, show how rethinking nutrient inputs and using food waste byproducts could reduce reliance on petrochemical feed additives and land-intensive crops. Raising protein with mixotrophic growth. The team, led by Yangchao Luo, Associate Professor, and Mingyu Qiao, Assistant Professor of Innovation and Entrepreneurship, applied a mixotrophic approach, combining light-driven photosynthesis with organic carbon feeding. By adjusting carbon and nitrogen levels, they increased protein biomass from about 25% to nearly 50%. Through feeding the microalgae with different media, different carbon sources, different nitrogen sources, and different minerals, we tailored their metabolism to produce specific nutrients, said Luo in the announcement. Replacing petrochemical feed supplements. The breakthrough carries major implications for poultry production, a cornerstone of the global protein supply. Chickens require methionine, an essential amino acid currently supplied through petrochemical-based feed supplements. High-protein microalgae naturally provide methionine, making them a promising sustainable substitute for chemical inputs. Beyond animal feed, the protein-rich algae could also serve as direct human nutrition, either as a standalone ingredient or as an additive in fortified foods. Turning waste into nutrients. Instead of relying on glucose, an expensive carbon source, the researchers used sodium acetate, a cheaper alternative that can be derived from food waste. With USDA support, the team is now testing acetate sourced from whey, a byproduct of cheese production, effectively closing the loop between food waste and protein supply. This proof of concept could turn food waste into a very valuable protein supply for human consumption or poultry feed, Luo explained. Why sustainable protein matters. Conventional protein sources face mounting challenges. Land and water pressures. Livestock and soy require significant resources, driving deforestation and soil stress. Climate impacts. Agriculture generates about 18% of global greenhouse gas emissions. Scaling livestock to meet demand could compound these impacts. Supply chain volatility. Reliance on petrochemical supplements and fish meal exposes producers to price swings and sustainability risks. Microalgae provide a scalable alternative, fast growth, CO2 capture, and compatibility with waste feedstocks. Global research suggests algae protein could eventually compete with soy protein concentrate in cost and sustainability. Funding and next steps. The project is supported by the U.S. Department of Energy's Algae Prize, the USDA, the Algae Foundation, and the National Science Foundation Future Manufacturing Program. Next steps involve scaling from lab conditions to industrial production and exploring strain improvements to maximize output. If successful, high-protein microalgae could represent a dual benefit, closing food waste loops while creating sustainable protein inputs for global food and feed markets. You've been listening to Sustainability Soundbites presented by E+E Leader. For partnership opportunities, reach out to a member of our team.